Thank you for having me. Can you hear me and see my slides?
Yep, everything is working.
All right, great. So, yeah, hello everyone. My name is Martin Helmkampf, and I just recently joined the lab of Oscar Puebla at the Leibniz Centre for Tropical Marine Research in Bremen, Germany. So most of the work I'm going to present on the genomic architecture of speciation in coral reef fish radiation was done by the former PhD student of Oscar, Cosmas Henk. And I want to start with evolutionary radiations, which are a major source of biodiversity, especially when they happen very rapidly. And on land, particularly on islands, we have several relatively well-studied examples of such adaptive radiations like the Darwin finches depicted here. We know a lot less, however, about how radiations unfold in the marine environment. And of course, we do have a lot of diversity in the seas, but we also have physical barriers and dispersal dispersal strategies that differ from those commonly found on land. And so to address this gap in knowledge, especially from a genomics perspective, we picked the hamlets as our model system. So hamlets are a group of about 18 species of small predatory fish living in coral reef ecosystems around the Caribbean. And what is remarkable about them is that even though they live extensively in sympatry, they differ very little in terms of their diet, their habitat, life history, and other ecological traits. They have, however, evolved into a stunning array of color patterns, as is depicted here, and they mate assortatively according to those color patterns. Now, in the last couple of years, we have collected a data set of 167 individuals. We have whole genome sequencing data of those individuals covering 13 species, and they were collected at 3 locations: Panama, Belize, and Honduras. We also have a chromosome level reference genome that we mapped those data to and have identified and phased 28 million SNPs, which form the basis of the analyses I'm going to share with you now. So the first question we were interested in is how those species are differentiated at the genomic level. So what you see here is a skyline plot with the chromosomes shown in white and gray, and on the y-axis the joint or average FST across species. And as you can see, genome-wide across species, we have very low level of differentiation, hovering just above 0. We also have, however, a couple of islands of very high differentiation that are highlighted here in with these red lines. And if you zoom into those peaks, what we found is that they are centered around a couple of genes involved in melanophore development, photoreceptor development, and similar functions. So we think we have a really strong case here of genes involved in vision and pigmentation driving reproductive isolation in the hamlets, mediated by assortative mating based on those color patterns I mentioned previously. A couple of other characteristics of this radiation. So one, it unfolded extremely rapidly. We have evidence for this in the form of demography or demographic inference estimates that estimate that all of this happened just within a couple of 10,000 generations, and also in the form of this older mitochondrial study by McCartney from 2013. The other thing is that gene flow seems to be ongoing within the complex. So we know this because we find in the field, we have observations of phenotypes or of hybrids of intermediate phenotype. And the SNP profiles also indicate that hybridization is going on, as you can see here with these blue arrows pointing to individuals that kind of sit between in terms of their SNP profiles in this PCA here. So as you can imagine, these are some really difficult problems to solve if you want to reconstruct a evolutionary history of the group, which is what we are still lacking. So this is strictly preliminary. What I'm showing you now, it's a neighbor joining tree based on 7 million SNPs. The branches are colored by species, and the wheel around is showing the location. A couple of observations here. So we see again that the hybrids that we have not removed yet from analysis are kind of falling into clades where we don't expect them to, and this probably decreases the resolution of the tree. We have a couple of populations that are not appearing to be monophyletic, like down here. This could be also a technical issue at this point, but we also have some pretty strong indications that some populations are actually not monophyletic, are in fact polyphyletic between the different locations. So it seems there's cryptic speciation involved in this complex and convergent evolution. So this is something we really need to look further into. So in summary, speciation in the presence of gene flow is facilitated by a very simple genomic architecture of species differences in the hamlets. We have high standing variation and effective population size, which I didn't show here, but this is something that is expected in the marine radiation. And these large effect loci generating phenotypic diversity and hybridization are now, it becomes apparent that this is a hallmark of both marine or many marine and terrestrial radiations. Now, what we definitely need is we need to improve our phylogenetic analysis, making use of what we would like to do, this population scale data that we have, and these challenges that I mentioned that are hard to overcome: incomplete lineage sorting, rapid diversification, hybridization, is something we need to address. And I'm hoping and confident that I'll get some inspiration for this during this meeting. We also would like to put this into the wider context of hamlets in the Gulf of Mexico and other surrounded fishes that have not radiated. So, and for this purpose, we have another almost 300 genomes in the pipeline that we hope will help us with this. Okay, I would like to thank our longtime collaborator, Owen Macmillan, and our funding agencies, the German Research Foundation and the Smithsonian Tropical Research Institute. And if you have any questions, I will have a poster number 12 tomorrow, and I'm happy to answer any questions. Thank you.